My name is Shelat Monson, the Stakeholder Engagement Officer of the Philippine Genome Center. We would like to remind everyone that we are also streaming this event live on our Facebook page. Please refer to the link shown on the screen. The live stream link is also posted in our Zoom chat. Please share to your friends if they are unable to join our conference through Zoom. As we may all know, this conference is divided into selected fields of application. And yesterday, we talked about current issues in health, such as functional genomics for dystonia, dermapathology and its contribution in developing targeted diagnostic kits and precision medicine. We were also updated with the current global efforts to understand, predict, and visualize the development of the SARS-CoV-2 virus. But for today, we will expand the topics to include important issues of global sustainability, such as food security, diversity and production in crops, livestock, and fisheries. To give us more information about the central theme of Day 2, it is our pleasure to call on Dr. Heidi Galvez. Dr. Galvez is an assistant professor at the Institute of Crop Science and former head of the Genetics Laboratory Institute of Plant Breeding, both under the College of Agriculture and Food Science in the University of the Philippines, Los Baños. She completed her doctorate in molecular genetics and plant breeding from the University of Melbourne, Australia. She has worked with the Fulbright as a Fulbright Advanced Research Fellow on Bioinformatics at the Boyce Thompson Institute for Plant Research in Cornell. And she was also a junior plant breeder at East West Seed. She has completed and is doing an ongoing research project on crop molecular genetics, breeding, genomics, and agriculture biotechnology. She was also the program and project leader of a 5-year coconut genomics program for PGC, which consisted of 8 component research projects. She has published research outputs and presented in peer-reviewed journals in local and international scientific fora conferences. Let us all welcome Dr. Heidi Galvez. Dr. Galvez, you may now turn on your video and share your screen.
Hello, good afternoon, everyone. So I'll be sharing my slide. So as mentioned earlier, I have been working in coconut, and that's actually one major program of the Philippine Genome Center for Agriculture. Hence, this I came to think of a nice opening for the Day 2, which of course we are tackling on how the different challenges and opportunities for bioinformatics and of course genomics as applied in agriculture. So we're talking of food security and of course health security for the future proofing in agriculture. So I'll just be presenting some initiatives and what milestones we have generated from the 5-year coconut genomics program and the opportunities that actually have been built, and how I'll relate it to how we have actually accelerated our capacity in terms of the expertise and in various applications in agrigenomics and of course for the Philippines. Again, good afternoon, everyone. I'd like to start my talk on sharing with you this inspiration that quoted from Nelson Henderson said, saying that, stating that the true meaning of life is to plant trees under whose shade you do not expect to see. So you can relate it as a service quotation, but for those who have worked in coconut can understand why this means a lot. It means that if you are working in coconut, you just provide the baseline knowledge pathway, but it will be too first or even up to 2 or 3 generations from your time, where you can really reap the harvest, as it needs long time for it to be developed into the final product, of course, which is an improved variety, whether you have applied and integrated molecular and biotechnology tools. So again, this the outline of my talk. So pretty much I'll start off with just briefly for your overview of the Philippine Coconut Genomics program, but I'll be focusing on Project 8, of which I'm the one leading this one, and some highlights. And then I'll be more, because we're talking on how from that coconut, we have the 5 year has all actually laid the foundation, more or less has helped the Philippine Genome Center, especially in application agriculture, how we have forwarded into different agri-genomics application. And of course, the tool, very important, and you know very well, bioinformatics. So I'll be presenting the different bioinformatics outreach we have forwarded from the program for the project, and then looking forward on what future research and data integration research collaboration we can put forward from the generated output from the project. And of course, here I'll be presenting the program, the Philippine Genome Center for Agriculture, R&D program, and some advancements and how we have surpassed the challenge during the COVID pandemic time. Of course, we're still continuing and still recovering and overcoming at the moment. And then, of course, I'd like to end to bring you back on what from the Coconut Genomics project, what we really envision to attain as our ultimate goal and outcome, of course, for the Philippine coconut industry. And this, of course, just laying the foundation, not only for coconut, but as a goal also for other PGC agri-genomics applications. So, of course, I'm the program leader here, but I am working with Dr. Reta Pilaude, who was really the first program leader who I had to take the role after she retired, but she's still working with us all throughout the implementation of the coconut program. So this is really a cohesive collaboration of with the Philippine Genome Center and of course the different constituent universities of UP. We have Los Baños, Diliman, and of course partnership with the Philippine Coconut Authority. And as already mentioned early on, it's a 5-year, and with phase 2 actually forwarding up to the pandemic time, 2020. So you see, even just from this period, you see that we're ending, when we are wrapping up, consolidating all data and all the materials. That's the time where we already start the challenges as imposed because of the pandemic situation. And we acknowledge very much the full support of the Department of Science and Technology through the Philippine Council for Agriculture, Aquatic and Natural Resource Research and Development, or from PICARD, for the full funding all throughout the program. So again, just presenting to you that there are 8 component projects. So project 1, which mean on coconut genome sequencing, and project 2 on marker development, project 3 on gene expression analysis, 4 another gene expression analysis, but for other traits, 5 just a proof of concept gene expression through genetic transformation system. And of course, we already start trying to apply some early developed markers in initial marker-assisted crop improvement. And here, also for genetic studies on trying to mine more genes, statistical association. So we did QTL mapping with project 7 and project 8, where I was heading, like from the genome sequence assembly, we tried to use it as proof of concept on mining genes. And in the case, because that's the time of the outbreak of the coconut scale insect. So we use as proof of concept to mine genes related or associated to insect resistance. So that's again a 5-year, started from 2014 and then 2019, and then some phase 2 projects forwarded up to 2020. So just to show you the overall of the program, so we cover really all the total genome analysis, sequence and assembly, so the whole total genome, nuclear or the cytoplasm, of course, chloroplast and mitochondria, because this is a plant system, and then of course the generated sequences. This is we really need high throughput computing capacity and expertise. So next generation sequence data. Later I'll show you as an example for Project 8, and then integrating knowledge on bioinformatics analysis to come up with the draft genome assembly. And then, of course, we have group also analyzing and sequencing, assembling those expressed sequences like transcript based on different characters. And then, of course, as in the phase 2, we are now integrating from the productive gene models from the draft genome assembly to validate those candidate genes with true gene evidence on gene expression data. And again, at the end, culminating for this sequence genome characterization and gene expression analysis is to come up with whole genome assembly of molecular markers. Okay, and then of course that's why we are saying that we for coconut project, it's really to just lay the foundation. Then the goal really is to make use of this genome-wide molecular markers for any trait of interest, quantitative or qualitative traits of importance to the coconut industry, to the coconut farmers, to the consumers, and all stakeholders. So then it really has to be used, all the markers for marker-assisted breeding or genomics-assisted molecular breeding in coconut. So again, just because it's you cannot visualize that you can finish in a 5-year project in coconut to come up with the variety developed from own genome sequence data generated at that period time of the project. But then we made proof of concept, as mentioned earlier, the draft assembly to my genes relating to insect resistance. And then there are already some early for yield and other qualitative traits working on marker development and QTL mapping, so that in a population, you can validate whether those candidate loci or genes or sequences and markers are really controlling or can really be a good tool as marker for in breeding to target those important industrial traits. Okay, so Project 6, as mentioned already earlier, is on initial marker assisted breeding already, making use of early developed markers. Of course, we have with us also from the Philippine Coconut Authority researchers. So now I'll just be focusing on Project 8, which is really on genome sequence assembly and making use as proof of concept on mining the genes relating or associated to insect resistance. So first challenge, so I already mentioned the challenge. The first challenge is a very long generation cycle in coconut, so that 5 to 15 years before you can see really you can have the plant or the progeny from the first cross, but it takes 15 years before you can evaluate for the productivity potential of the crop, because it for dwarf, for example, varieties, 3-5 years can start the flowering, fruiting, but it's not yet the peak of the production. For tall varieties, 7 years and so on, but you need more additional years to really see the peak of productivity. And then another challenge is it's really a very large and complex genome of around 2.4 gigabase in genome size, and very rich in repeats. And for those who have worked in genome assembly, you know the challenge that is really imposed for this project. And I can share that it was really ambitious for the group. Says it's really our first whole genome sequence project of course for the Philippine Genome Center, especially for application in agriculture. And we started with just a very challenging organism, and that is coconut. But you'll see that we have got uh, good accomplishments as like baseline for follow-up research studies and work towards genomics assisted crop improvement in coconut. This has been just small efforts or accomplishment from each of the different projects, but if you sum them all, and of course, as we have to continue to catch up with follow-up researchers, it will really give an impact, and we hope to achieve what we aim to as the ultimate goal in coconut crop improvement. Okay, so first, of course, already I mentioned the challenges, but and then our how we say our courage to start with a very difficult crop for our first whole genome sequence assembly project, but we made it. We were successful because of very good tied up and collaboration with experts already in the field. So we know that it's our first time to work on this, and as we have to capacitate ourselves, we have to deliver because it is a project based, and we have a deliverable to the funding agency and of course to the coconut industry. So we work with the Boyce Thompson Institute for the whole genome sequence assembly in Project 8 through Dr. Lucas Mulier Group. So there were students and staff directly working in the project. So just short internship, and then after that, we are working on our own back in the Philippines, and then we just continuously getting consults and expert advice from them virtually. So we actually have a graduate master's student who really did the whole as his master's thesis, the genome assembly of as output, main output of Project 8. And then of course, with the other projects, we have also to work, this is again for genome assembly, more on whole genome and some transcriptome analysis. So with projects 1 and 3, they have to work and get also expertise from the genetics, National Institutes of Genetics and Shizuoka, Japan, and all the technical consultancy. And there were also shuttle and exchange of staff. And of course, with Project 4, again, this is for transcriptome analysis. And they work also closely with the Pennsylvania State University through Dr. John Carlson. So technical consultancy and of course, expert on sequencing facilities, and also access on some sequencing facilities and bioinformatics analysis. And then I'd like also to mention that Dr. Lucas and Dr. John Carlson are actually a member of the International Scientific Advisor of the Philippine Genome Center for Program for Agriculture also. So they have been helping with us for this endeavor. Another very important material that has helped us really to advance is, as I mentioned, the generation cycle in coconut is really very challenging, and 15 years before you can have your first generation adequately, you can evaluate for productivity traits and other potentials. We are so lucky that the Philippine Coconut Authority has already materials for us. The germplasm, well characterized in phenotype and other preliminary marker, molecular marker characterizations, and they have a genetic map populations that are really designed to evaluate for target quality or yield traits in coconut. So at least there is already a 12-year-old fruiting stance where very baseline initial preliminary study, but can be done and adequate to start up preliminary study on QTL analysis. And you can actually, because they are already fruiting stance, so we can already evaluate for the yield potential and other qualitative traits. So, okay, so with Project 8, our goal really is to construct the genome-based database for coconut. And then we hope that with our collaborators with Boyce Thompson Institute, we can use as our backbone as prototype for the database, some pre-installed tools for breeders. And then, as I mentioned, we will use it as proof of concept to access the assembly for mining genes and then candidate genes and markers, proof of concept targeting glandular trichomes, scale insect resistance, and of course, we will be mining those potential alleles and other variant genes in the germplasm collection with our partner from the Philippine Coconut Authority. So just to show you the overview, so we have different research activities for Project 8. So we have actually 4, and so there are many research leaders involved and researchers, project hire and regular staff of UP. But just to show you the overall framework, so of course we have the study on genome database assembly, sequence and assembly, and then of course tied up with targeting candidate genes and developing of markers on those gene sequences. And then of course there's also our entomologist group to work with the insect pest. This is the coconut scale insect. So molecular validation and characterization. And then of course, as mentioned, those candidate sequences, we already start mining potential variants and interesting alleles or genes from the germplasm collection of the Philippine Coconut Authority. So with that, then we can have some already candidate markers targeting on those candidate gene variants for insect resistance. And of course, we can already have tagged some potential tolerant, if not resistant populations or varieties or genotypes of coconut. And then, of course, along with this is we need to do at least greenhouse and on-field actual infestation and field trial for host resistance screening, since we're looking on the trait, validating those candidate genes based on predictive gene models. So we see also reaction in the field, in the greenhouse, and of course, as we correlate it, insect resistance, so trichome, glandular trichome. So we do also trichome characterization of this identified germplasm of coconut. So just to show you the overall workflow of the bioinformatics we use for the genome sequence assembly in Project 8. So of course, at that time, 2014, 2015, PacBio, and then of course with it started with Illumina and then came PacBio. They were the advanced sequencing platforms at the time. And then towards us, we have already built the draft. Then towards the finishing of the draft assembly, then came also introduce of the advancement in dovetail Chicago sequencing. So with our collaborator with Boyce Thomason Institute, we were able to get access to this sequencing facility and obtain some adequate row reads where we use to improve the draft. And then with applying the best available programs at the time, of course, also an assembler, we able to come up with our draft genome assembly. And then Project 8, we did not do actual gene expression analysis or transcriptome data, but we made use of all available sequence data, transcript data, gene expression data in the database, and applying again all the appropriate bioinformatics tools and program. Then we came up with the annotation of our draft assembly for some gene models, and of course, characterizing functionally annotated genes. And since, of course, we're interested on insect resistance, then we look on evolutionary analysis of identifying candidate resistance gene analogs in the whole coconut genome. And of course, along with it, characterizing some drought-related genes. And since this is coconut, we also already characterize or tag those genes associated to oil biosynthesis. And of course, even without the transcriptome data or annotation, we already have predictive and identified genome-wide markers that can be used later for any breeding application beyond what the data can be provided during annotation, since you are limited on what's available in the database. But we have already markers whenever there are evidences and gene expression data that we can use to validate any of this as our marker. So that's one major output as well of the project. So again, just some highlights. So we did came up with a draft and good quality genome assembly and annotation of course, we work with as our first reference genome, a dwarf variety. This is a Philippine Katigan green dwarf coconut variety. And then, as we said, we have already a prototype genome database in the backbone, same as used in Solanum Genomics Network. And we have already ready pre-installed relevant informatic tools and plant breeder tools also. So we actually have a publication of this where you can refer to for the details of the output. And I just highlight some important results from the genome assembly analysis and characterization. Like, for example, we at that time or for the data we have adequately covered 2.1 gigabase of the genome size of CatD of Katigan green dwarf. That's around 98 coverage, and staples of 8,000 with 569 KB and 50 quality score. And then genomic simulation, as we analyze it in as comparative genomics with major plants. So, for example, we found 3 rounds of whole genome duplication events in comparison with all other Aricaceae. And very important, of course, because we are more into the application of the genes and marker development towards crop improvement in coconuts, especially for economically important traits. So we take note of protein coding genes, transcription factors, and of course, transcriptional regulators. And since we are later on will really be very interested in using any of these target genes and markers for resistance breeding or application for coconut improvement, then we take note of resistance gene analogs and drought response genes, and of course, major genes for oil biosynthesis in coconut. And of course, I just did not elaborate here, but you can see the paper, and there are all other genes or any other genes of interest needed to improve the coconut industry, production, quality, for nutritional and even towards health application. So we have already genome-wide DNA markers that can be used for molecular breeding, whether for climate change resilient development of a variety, for pest disease resistant coconut variety development, of course, for high oil yield and improved quality. So just to show you we have identified and marked genome-wide markers that can be used for high throughput application. And of course, since our entomology group also work with the coconut scale insects, so we have a species-specific DNA marker developed to differentiate the different species of coconut scale insect. And this is based on our initial field trial for natural infestation involving all available and pre-selected coconut varieties. So we have identified some least damaged coconut varieties under natural field trial conducted with the Philippine Coconut Authority. And then since for the resistance gene analog, so we have characterized as we are looking on insect resistance, those genes that are related, associated to trichome trait. And then we use it to already mine allele diversity or gene diversity in those identified PCA germplasm, just to correlate also those candidate genes as well as what we observe on the actual field trial. So we have marked, identified those genes and alleles that may be associated to host resistance directly or indirectly based on association to trichome trait, to glandular trichome trait. So we hope that through forward and reverse genetics and correlation studies, coupled with relevant genome and bioinformatics data, the mechanism of host insect resistance will be elucidated in selected coconut varieties that we have identified, and there may be other varieties that will be have to be screened and identified. And then, of course, I mentioned that since coconut is really a crop for a source of oil, and we also have identified already characterized all genes related in the coconut genome. So I'll mention that I'll be sharing you the bioinformatics workshop that I would say a major outcome also from the project. Since you will note that it's really an 8 projects and a strong collaboration and joint effort of the different UP constituent universities, and many of our project staff are already employed already by either from the different universities and even involved in other state colleges, universities, and some of the project staff also from the coconut are the one also involved in the other PGC uh, facilities in Visayas and Mindanao. So there were really 3 major bioinformatics workshops throughout the duration of the project, and we work it with our collaborator with the Boyce Thompson Institute with Dr. Lucas Miller and his team. So first was really just introduction on Perl scripting and as beginner biologists, students, and researchers. So that's with UPLD. And then you'll see the progress also of the learning. It's actually 1 year after as well, when we are starting already having our own data. So we have some data to look at, and then of course we have a training data set for us to work on as well. So the second bioinformatics, again involving mainly the project researchers from the coconut, and of course all other researchers and students that are working in genomics and bioinformatics. The second is already on variant calling or SNP calling with using an existing next generation sequence data. And then of course, when this is already towards also the end of the project, when we have already the draft, some of the raw reads being analyzed and now building the draft genome assembly and running all the modeling and annotations. Then we had alongside, as we capacitate ourselves, we also conduct and open up that bioinformatics workshop to the researchers and students working on genomics again and bioinformatics in the university. So the third bioinformatics workshop was on genome database development and gene mining. So and then of course we continuously provide genomics lecture and bioinformatics workshop beyond the university or beyond the project. And I'd like to mention here, I guess I missed another university here. We have really also a hands-on training workshop with all the research centers of our partner, coconut industry, the Philippine Coconut Authority, with Zamboanga Research Center, Albay Research Center, with Davao Research Center, and of course, they have also representative from their R&D branch from the main office. And also we work with from representatives from DA in Mindanao. And of course, I forgot to add here, we also had some hands-on training also with in Visayas, with Visayas State University, with the Coconut Research Center, with the Coconut Research Center. So of course, again, introduction event to the high school student. Here, as part also of one major output and I would say outcome, beyond the coconut project, we have one of from our Institute of Computer Science, Prof. Aryan Hasildo. He went for his part of his PhD experience also that he did preliminary of his dissertation with the Boyce Thompson Institute in the same laboratory, and then having those exposure on how to merge a hardcore computer scientist now working in real biological data and real biological problem that have really motivated and inspired him more to go forward and apply computer science in real biological application. So of course, beyond that, we have a lot of initiatives already and publications working with other biological researchers. So that's, I would say, a very good output. So in summary, from the project 8, we have generated the draft assembly of coconut genome, specifically from the Philippine Katigan Green Dwarf variety, using several sequencing platforms. And we have also sequenced the CSI, characterized and have developed species-specific DNA marker system. And then employing a preference or test in the field, we have identified some least damaged coconut varieties and have mined those candidate genes, the variant genes in different germplasm populations. And we hope to forward this further as we are looking to have our further research activities on understanding the mechanism of insect resistance and more towards the construction really of the coconut genome database in integration with other available data and consortia so that we really attain the aim of making it available to all coconut researchers that don't need to be really in the hardcore bioinformatics expertise. And of course, continue gene mining and marker development. And again, we work to have more populations to be sequenced to have a good depth coverage and towards more advanced application in gene association, characterization like GWAS and whole genome sequence analysis for other genes. And of course, I mentioned already the importance of drought tolerance since coconut is very susceptible when the water is very limiting. So it is always a limitation during productivity when there's drought. Our farmer may not have any harvest at all. So of course, the farmer don't have an income, but the coconut industry will also be because they rely the supply from the producer. So all the stakeholders of the industry will be affected, including even our export market and takers. So of course, also drought genes have been identified. So here we have now a ready assembly of our whole draft genome sequence with the markers and data. So we hope really to work further and integrate it with the existing genome database so that, as mentioned, this becomes made available for all coconut researchers. So I'd like to acknowledge here all those workers in the coconut genome projects. But before, let me have this as my breaker slide that we have just starting. And we hope that we continuously progress and continue working so that we will attain the goal of the application of genomics in agriculture. So now I'll just be just quickly introduce to you also the all other applications of genomics and of course bioinformatics in agriculture. So I'd like to, of course, this is already presented. We have the Philippine Genome Center as R&D program in agriculture, livestock, fisheries, forestry program. And then, of course, this is the organizational chart of the PGC in agriculture. So we have here a very dynamic targets for different commodities, crops, animals, of course, fisheries and forestries and microbial and arthropods. And we have the functional discipline as our functional group from genomics, transcriptomics, metabolomics, proteomics, epigenomics, bioinformatics, and of course, since this is really towards molecular breeding application or preliminary QTL. So we have, I'm actually presenting this on behalf of Dr. Dr. Tony Lorena, the current program director for agriculture, fisheries and forestry. And then of course, we have now our core staff. They are full-time hired for the PGC Agriculture. And you will see the distribution. We have a lot in percentage MS level graduate. We have PhD and we have just new staff of BS. And of course, we have all here already for the different functional disciplines or group of the PGC Agriculture program. So just browsing through the completed projects, of course, I have here coconut, but there are many other crops that have already succeeded to try apply agriculture genomics tools and bioinformatics in various agricultural commodities and different molecular tools involved. So in crops, livestock, fisheries, forestry, and of course we have also microbes, because in agriculture we work with the microbiome also in the ecosystem, rhizosphere, and even within the agricultural system. So ongoing researches, again, cutting across the different disciplines and the different commodities. So you see that we have really advanced in crop, of course, in animals, or in application in livestock, and in forestry. And then, but we have already in the proposed projects already to include application also in microbial and arthropods. And so far, in terms of what have been accomplished, and I'd like to note that this is cross-listed with research projects as spearheaded by various UPLB academic research units. So this is not like just solely done with our core staff in PGC Agriculture. And you will see later that with those few core staff, but they have really a wonderful output. So in publication, I haven't really gotten to count. It's just the report I checked as of December 2018. So there are more than 11 published manuscripts. So the early projects in pili, shrimp, coconut, coffee, tomato, but there are several publications that have now been submitted or drafted for submission to scientific journals. And you will see that many things have already been written during the pandemic, when we have a lot of those challenges in continuing to work in the laboratory or even to report to work. And a lot of virtual, because it's again during a lot of the outputs during the pandemic time, oral and poster papers in various scientific conferences, and for including international, which have been done virtually. So of course, for patent and some registration, there were initial patent identified and tagged for PCR-based meat identification, and of course in coconut for genome-wide coconut marker. For products, there were many. So from coconut, then we have seed nuts, from genome sequence assembly to marker. Of course, we have initial transformation protocol and seed nuts that have been harvested and seedlings distributed. In other crops also, we have candidate varieties in sugarcane for high sucrose content and for downy mildew and smut resistance. And of course, in OC red cross, there's already an F1 progeny, then a cheese recipe for native cattle milk, gene markers for various commodities for high yield, resistance tolerance, and other quality traits, microbial population, those collection from Mount Pakiling, so been characterized. And then, well, because we have now the ongoing project on genome editing using CRISPR-Cas9 as the early technology. So we have some gene constructs in tomato, corn, of course, in rice with our partner, and then tissue culture regeneration protocols for, again, in the CRISPR gene editing project. So as partners, it's really challenge to work in challenging and complex commodities, different from crops, and livestock, forestries, and then of course the in marine or fish. So we have different partners. So I mentioned already our partners, which we helped us a lot to come up with those very good baseline accomplishments to forward in coconut genomics breeding. So we have with Boyce Thompson, we have with National Institute of Genetics in Shizuoka, Japan, and Pennsylvania State University. But we have also with Iowa State University through the USAID Stride project in the cattle project. And for the shrimp, we have National Sun Yat-sen University in Taiwan. In Taiwan. And with our continuing projects, we have worked with a partner also, of course, with IRI, International Rice Research Institute, and with National Research Institutes, with the Philippine Coconut Authority, with PhilRice, PhilFIDA, some industries and industries, and recent with Dr. Angeles, where he did genetics training for in 2018 with Dr. Chris Tazzonelli in Western Sydney University. And we do have strong cohesive R&D projects, which we partner with the other PGC R&D programs, like with health, biodiversity, and computational programs, R&D programs of PGC, whether project-based or core research efforts. And of course, with Miss Julian, she went for internship also in Iowa State University with the Department of Animal Science for some internship with Dr. Juan Fernando on animal GWAS as part of the Cato USAID Stride project. So again, just with another more partnership for capacity building in bioinformatics. And again here for genome editing, we have Dr. Diaz. She went for hands-on training sometime in October, just towards the end of 2019. Before we had the start, of course, we have also the Taal. If you remember at UPLB, we have also the Taal eruption on December, some tremor already December, January. And then when we were, I have a wrong spelling here. We were already towards trying to recover from the some eruption, then came the pandemic. But before really ending, we even had a hands-on training with Dr. Riamilio also went for some hands-on training also on genome editing with CRISPR-Cas9 in UC Davis. Also, again, towards last, before the pandemic situation, like from September to December of 2019. So in policy, we have position paper of the PGC Agriculture in response to House Bill about GMO Labeling Act. And of course, there's the turnover to PCA. There's also policy of those individual farm selections for legal protection as on-farm genetic resource, standing tall, and those identified in the field as SI survivor trees. And very important from the learning and some materials we have with CRISPR Genome Project to contribute to the Philippine Biosafety Guidelines policies on the use and handling of gene editing technology. So in terms of people services, I guess we have really supported a lot of students. We have already identified those who have graduated. So from BS, especially BS Biology, Genomics, working in genetics and mold bio and cellular biology, and of course also in BS Agriculture Biotechnology students for their thesis and some internship, also master's students, and of course PhD students. So we have more 62 students already graduated from us, and of course several bioinformatics hands-on seminar workshop conducted whether project-based or organized by the PGC Agriculture course staff, and several other technical trainings and lectures. So before the pandemic, face-to-face, and then a lot already done virtually during the pandemic time. Example on beekeeping, genome editing, and of course there's regular journal club and seminar series continuously being hosted, organized by the PGC Agriculture course staff. And we have scientists trained on CRISPR-Cas9 genome editing. So awards recognition, too many to mention, and I don't want to mention one and then I fail to mention those that I have not checked. So there are several poster oral presentation awards in different scientific conferences and fora, and several paper research and achievement awards of either our UP, different UP faculty researchers, staff that work directly or to any of those genomics project of the PGC Agriculture. Okay, so how those have been accomplished. So I mentioned that there are only 9 to date, the core staff of the PGC Agri, but then we have a lot of some UP staff that actually many of those are holding multiple projects. So there are just count, approximate there are 36 involved project personnel, including UP staff, that in distribution, we actually have only few PGC Agri core staff or full-time. And then we have UP and project hire. So really, since these are a lot of project-based genomics research, so we managed to hire full-time project staff based on the funding from the different projects. And of course, as I mentioned, a lot of our expertise from regular UP staff that actually have to hold multiple projects. So hope this we can know. And then in terms, of course, of project funding, again, this is just as of December 2018. We have I just consolidated. We actually have this is again including cross-listed projects that directly that don't have to be directly managed here at the PGC Agri administration. So there's a total of already 442 million and 803,682 around pesos that project funding. And as you see in distribution, still a lot, since of course there are advances in crops. So we hope that we will leverage also advancement in the other commodities. So come, because I have to open up the day for how we have turned those challenges during the pandemic to opportunities. So I'll just show you that we have ongoing projects that really cut and continuing and being captured with the challenge situation. What specifically the lockdowns, community quarantines. So we have Coconut, the phase 2, and some wrapping up of the completed 5-year project in Abaca. In of course our new breeding CRISPR-Cas projects involving tomato, corn, and rice commodities, different crops. So those lockdowns, different levels. You can have quarantine, community quarantine that can be strict and radically can be changing. When everyone knows that, have really hindered the travel of or restricted the travel of employees to workplace. We all share this one, restricted sometimes, aborted experiments, whether already ongoing experiments in the laboratory, in the greenhouse, or in the field. And if you have already a program, scheduled travel or conduct of off-site experiments, and we can actually share our supposedly continuing hands-on training on CRISPR with our staff at Erie, which was aborted or put on hold, suspended. And of course, this have direct impact on the project implementation, on, of course, delivery of the output. But indirectly, the delayed and suspended administrative transactions, because again, of these lockdowns and community quarantine, there was a shift of usually a physical processing of documents and then migrating to online virtual online submissions, but yet there are still hard documents that have to be processed as physically office to office as hard documents because of some auditing requirements. And then of course, that delay not only some of the project implementation, but especially even the delivery of supplies or services. So you can have already an approved supplies already ready for delivery, but could not be delivered, or approved contract for repair or some services that could not be conducted or delivered by your supplier because again of this pandemic situation. So it's 2019 to 2020, then towards the end of 2020, we're trying to catch up. The hard lockdown was really March, was it? March to almost August, July. But then how we managed to overcome, actually when the pandemic already starts to loosen up, at UPLB, everyone just really helped along, and with strict guidelines, we follow strictly the guideline from the, of course, relayed from IATF and the national government, the local government, and then with the memo released from the university. So we experience with 10%, 20%, 30% workforce, 50% work from home, and some face-to-face. So it depends really on the stage of the critical stage of ongoing projects. So there are situations where 50-50 of your project staff have to report, and half is in the laboratory, for example. So you make sure that you wrap up, and then when the other 50 remaining project staff would be reporting the remaining days of the week, you have to make sure that there's a good transition to pick up and continue the work. So it's like a relay, 50-50 relay also of to be able to really meet and deliver on what has to be delivered in that quarter for the project. There are also some experimentation, like you continuously, those 50% continue for the first 2 weeks, and then the next 50 just to make sure that there's a continue conduct of the experiment up to a point where you can stop and can be picked up without problem for the next researcher or for the continuing conduct of the experiments. So it depends on the projects, and it depends on the need of those experiments, whether in the laboratory, greenhouse, or in the field. And for the administrative like documents and so on, I guess patients of all were really tested and trying to help everyone. So it's like whether you are a technical, you really have to work also on start learning some of those paperwork and administrative transactions. Since of course we have to support also the central administration, since everything will really just backlash to how we can really implement the project and be able to deliver. I guess the clock there when we submit reports and requests for renewal, of course the funding agencies are very considerate, but the clock is still ticking, and especially with living organisms like plants, and you cannot stop. There was a situation where I have a tomato plant in the greenhouse already fruiting and ready for evaluation for phenotype and for biochemical characterization, but there was a hard lockdown. So what we just did is just have that one technician assigned, harvest it, keep the seeds, and that's it. So at least we have the seeds for the next planting, and we can do the another round of planting and evaluation. And in some situations, we have to send the seeds, and our students, especially those catching to finish the masters, have to grow the plants in their house and do whatever analysis they can do just to generate the data. But in situation where bioinformatics, they're already in the data analysis part, bioinformatics, then I guess that becomes where the time they can conduct even remotely from home. But the challenge again is of course the capacity, the internet connectivity, and the capacity of the computer. But everyone just adjust and overcome those challenges and came up with reasonable output given that period of time. Until now, we are still trying better situations already, but still there are a lot of hurdles, challenges still of those two major points, either on lockdowns and community quarantine, and of course still on administration. So I just summarize them into those two major points. I'd like to mention this as one hard hit, especially on our project in tomato, where we had scheduled in 2020 to 2021 a research fellowship of 2 PhD dissertation. This is for 1 year. 2 PhD students, they were awarded already for a 1-year research fellowship in World Vegetable Center in Taiwan. They would conduct, supposedly would have conducted heat tolerance GWAS in tomato, while the other one heat tolerance GWAS on pepper. And then of course, since this is in this is for our CRISPR project in tomato, we have our Greek scholar. She was also awarded supposedly with a research fellowship for 6 months to conduct CRISPR experiment in tomato in their laboratory. Again, this was supposedly on 2020 and 6 months in 2020, and of course the PhD dissertation 1 year 2020 to 2021. And there's also part of the MS thesis research for the 6 months, a short research visit, I guess 2 weeks, as I recall the calendar, for a 4E visit also to learn hands-on and do project coordination to initiate future projects with the World Vegetable Center. So all these were canceled or suspended. And then I have here how we mitigate, because of course they have to graduate. So we made use of bioinformatics. So the PhD student is currently working now for her thesis with ERI existing data to run bioinformatics analysis, while the other PhD student also with existing data to run more data analysis on QTL analysis in court. While for the MS thesis student, she graduated with all those hurdles. So we maximized the bioinformatics on preliminary design of target regions for gene edits in tomato and gene construct assembly for the guide RNA and so on, and then some baseline molecular genetic study. Those I remember, if you noted, I mentioned the tomato that she had to bring at home to continue some of the basic molecular genetic study just to make the whole story of her thesis. So she graduated with all those hurdles just in time for the requirement of her DOST scholarship as a Greek scholar. So again, for the short research visit, there's again another opportunity. So it was not, it was canceled or suspended, but then there comes an opportunity for a Fulbright research visit scholarship also, still on tomato genome editing, that will be next year. So just short 3-4 months as well. So again, I use this as also another opportunity that we have for during the just this year. We have new staff to add also to the course, agriculture staff of the PGC. So we have for the molecular breeding, and we have also for the genomics transcriptomics. And so that add up the contribution of our course staff here and their distribution. And then we have also during the time where we have again a realization as during the reflection during the pandemic time, we have one project staff, PGC course staff who resigned. So there is again as opportunity and available position which we are, I guess it's already called for filling up, and there are already applicants for evaluation. So I took it as an opportunity. And then of course, another opportunity for the capacity building of the PGC course staff. Of course, Dr. Zaportiza is currently just when the international travel has loosened up. So again, this is in connection to our CRISPR project, so in court. So Dr. Zaportiza is currently in University of Hawaii for her Fulbright research also, conducting genome editing in court. So when she came back, then we'll continue it in the project, and there will be a follow-up research also project being proposed with the funding agency. Another opportunity, just I guess she Miss Julian Villela just March, somewhere during March or just before the really border closure of international travels, Miss Villela was able to travel to UK to do her PhD, on molecular biology in University of Lancaster in UK. So she's actually working, which is very timely also. Her dissertation research is on vaccine development for virus disease in livestock, poultry. Okay. Okay. And another opportunity I would say that also still happened just to give us some light and for the pandemic time is we have one staff, of course, that has found her Mr. Right and having a new member of the family already. So that's very welcoming as well, in spite of the situation. One thing is, you will see here, there was already a plan, a very defined milestone to put up the PGC Agriculture Central Laboratory. And you see that supposedly this is the construction time where we are hard hit with the situation of the procurements, document processing, and even the construction itself, and even to the supplier or constructor. Then that's one, the challenge. And then of course, this total building plan as proposed would cost around 120 million pesos. But of course, we only have initial budget just to lay the foundation. but at least this is already crafted as up to the finish completion to have a functional central laboratory for genomics application in agriculture. But then the current budget is for laying the foundation, and then taking the opportunity again, because there is a whole package already of the needed documents, and for us for the preparation of that proposal for the building, there's opportunity now of the COICA project, a UPLB COICA ERA project for capacity building for higher education and establishment of UPLB Agriculture Genomics Center. So this is a 6 years project, and actually more or less officially will be starting this year up to 2027. The main goal is to construct the UPLB Agriculture Genomics Research Center, or UPLB AGRC, as a COICA project donation to the Philippines through UPLB, and then of course provision of the needed equipment to be able to conduct agricultural omics research. And then of course, since you have the building and the facility, capacity development of the personnel and students that will be conducting agricultural genomics and all other omics applications. So it will be with Korean universities and also with expert assistance from ERA scientists. So there will be full scholarship for PhD and dissertation research grant support for thesis, PhD, masters, and of course undergrad as well, training, internships, practicum, and seminar conferences. And very important is there will be a part of the project is to have co-development of UPLB curriculum applying agricultural genomics. So that was the plan, and then suspended because of course limited budget at the time, and then because of the suspended again because of the pandemic, then come the opportunity. So we will have the UPLB Agricultural Genomics Research Center, and we have actually just recent, we have the PGC Agri group have with the project leadership of Dr. Jenna Diaz, have an approved equipment grant as augment also to the COICA project from Picard DOST. So during another major accomplishment, I would say, or how we took the opportunity during the pandemic time, is to have reviewed and really laid down the strategic plan or roadmap of the PGC Agriculture way beyond 2020 up to 2035. And that was actually the takeoff from the strategic planning workshop with the Deputy Director, Dr. Raul Distura, which was conducted just before the lockdown, I guess. Well, that was on March 14, 2019. That was conducted here at UPLB. So it was time for the group, for the PGC Agri-Core staff to really review and plan so that you cover, so way beyond 2020 to 2035, to cover not only in crops, but livestock and forestry, and of course, livestock and fisheries, and of course, forestry as among the commodities in the program. Humbling experience and opportunities, a real life expert. Yes.
Hi, ma'am. Hello. Hello. Good afternoon.
Yes,
John Michael Yap here. Just gonna ask you to wrap it up na po para mabigyan naman po natin yung mga speakers para
Okay, ito na po yung Sige, thank you. So just to show you that directly we have the UPLB efforts participation on COVID-19 pandemic response. So as members of the R&D committee of UPLB COVID-19 task force, members in the committee for the establishment of the COVID testing center in UPLB, and then of the different projects in support to the COVID-19 pandemic response of UPLB, and this one especially for Dr. Diaz and Ms. Grace La Chica to be members of the committee for the DOH validation of the biotech as become the converted UPLB COVID-19 testing center laboratory, and then have supported the project involved in genomics, supported the efforts also in establishing the laboratory by loaning important equipment like that can be used, and also laboratory supplies. And to start off the laboratory, the COVID molecular diagnostic lab at UPLB, we actually started volunteered project staff from genomics project, and then now they are employed as regular staff. And of course, we have now Dr. Diaz as the laboratory manager of the COVID testing lab of UPLB, and Miss Grace Lachica as continuously working as the assistant laboratory head for the operation of the. And then there are still current projects for and still here for the opportunistic outcome project to be implemented from the response of COVID response effort of UPLB. So with this, this is actually what we have. We hope that with to have more support for the increased core staff. This is the envision plan of the PGC Agri core research staff, and with the coming reinforcement and availability of a genomics research facility, that hope we can achieve for the outcome of application of agrigenomics, genomics and bioinformatics in agriculture. So I'd like just to end as an example, the outcome that we hope with the Philippine Coconut Authority, with the coconut genomics program is really to address the global challenge of local and global challenge of the production, as well as, of course, the coconut oil, and to address the coconut farmer situation and maximize the potential products and that can be derived from all parts of the coconut, and addressing beneficiaries towards helping the farmers, supporting our researchers, the industry, with the Philippine Coconut Authority, and all stakeholders involved in the coconut local and global. So with that, I guess that's it. And again, we hope that just small efforts repeated each day will give us really the great success in genomics and bioinformatics application in agriculture. For that, thank you very much for your time and listening. Thank you.
So much for that very comprehensive update of the PGC Agriculture program, especially the coconut genomics program, Dr. Galvez. We wish for further updates, hopefully with more support from the people you reach out to. Now, please take note for our participants today that you can also submit questions using the Q&A tab during the speaker's presentation. The Q&A tab is located in the bottom part of your screen for desktop, and the upper right corner of your screen when you're using your mobile phones. Now, to get into it, our first guest speaker for today is Dr. Nelzo Ereful from the National Institute of Agricultural Botany, University of Cambridge. Dr. Ereful is an experienced molecular biologist and bioinformatician with a strong interest in molecular evolution using high-throughput sequencing technology and computational statistical packages. He has a strong interest in using crop hybrids to identify candidate genes associated with stress tolerance using both molecular and computational tools. To let you know more about it, he will be presenting his, he will discuss RNA sequence reveals candidate genes associated with high fiber quality and bunchy top virus resistance in abaca. Dr. Ereful, you can now share your screen and your presentation.
All right, so good afternoon. I think I've sent a video.
Good afternoon. My name is Nelzo Ereful, and I'm here to present to you our work on abaca. This is entitled RNA-seq reveals candidate genes associated with high fiber quality and bunchy top virus resistance in abaca. Musa textilis ni. And I would like to thank Dr. Antonio Lalusin and Dr. Laurena for their help and support during the performance of this study. The objectives of this study is actually enclosed or included in the title, which is to identify candidate genes associated with high fiber quality in abaca using RNA-seq, and identify candidate genes linked with Panchytap virus resistance in abaca. The methodology of this project includes the collection of 3 abaca varieties, including Abuab, which is recommended for Luzon, Inosa for Visayas, Tangongon for Mindanao. A wild indigenous banana variety or a Musa balbisiana variety Pacol was also included, and 2 backcross genotypes, BC2 and BC3, which is a cross between Abuab and Pacol, were also included. These were all grown and maintained at the Feeds and Industrial Crop Section collection site at the Institute of Plant Breeding, University of the Philippines, Los Baños. And the central whirl of the stocks of the plant materials with uniform growth status 3-month-old were collected at the site. The site is here in Luzon. And we collected 3 biological replicates of the 6 varieties. All of the 3 biological replicates were pulled to make up to 1.0 for each of the variety. So that means there are 0.33 grams were extracted or weighed from each of the biological sample, and they were pulled to make up 1.0 grams. There are 6 variety samples in all, which are, all of the samples were extracted for RNA sequencing using modified CTAB method. To give you a background on the back process, the BC2 and BC3, Abuab was crossed to Pacol at the initial stage or the original stage of crossing. Abuab is the female parent recipient and Pacol is the male. In the succeeding back process, Abuab served as the recurring pollen donor, the source of high fiber quality alleles, while Pacol is the source of the Panchytap virus resistance. Note that BC2 is called Bandala, which stands for backcross abaca with native and desirable accessions to lift up the abaca industry. This is further crossed to Abuab to generate BC3. So only BC2 and BC3 were included in this study. The phenotypic performance of the 6 different Musa varieties is tabulated here. Abuab, Inosa, and Tangongan are all susceptible to abaca Panchytap virus. The backcrosses and Pacol are resistant to abaca Panchytap virus. The persistence of the backcrosses is conferred by Pacol. And Abuab, Inosa, Tangongan, and the backcrosses have all high fiber quality, while that of Pacol has a low fiber quality. The bioinformatics pipeline we have implemented is that the FASTQ files were checked for quality, and fortunately, there are no adapters and there are no low quality bases, so there's no need to clean because they are very clean. The abaca varieties were mapped against the Abuab reference sequence, which was recently decoded by Galvez et al. In a recent publication, while that of Pacol reads were aligned against the Musa balbisiana double haploid Pisuklutokulung, and transcript orthologs were searched between Abuab and Pacol, because abaca and Pacol are interspecifically divergent. We look for transcript orthologs using the software Orthoven, and the reads were aligned using star, and aligned reads were quantified using feature counts. And we performed differential expression using BASEC with normalization procedure to correct for segment length and sequencing depth. If you want to access the different RScript and Linux commands, they are in here in this GitHub container. The results of multidimensional scaling plot explains that there is divergence between Musa textilis, and Pacol, which is Musa balbiciana. And dimension 1 explains 53% of the variants. So you can see that Pacol is on this extreme right side, and Abuab and its back roses are clustered in this quadrant, while that of Tangongon and Inosa are clustered in the lower section or quadrant. Pairwise differential expression between Pacol and each of the Abaca varieties show that Abuab and BC3 have the highest proportion of upregulated Abaca genes. So the performance would be very good in terms of fiber quality, which means further that these two are the most adapted varieties in Luzon, because this study is performed in Luzon. BC2 has relatively very low upregulated genes as compared to the downregulated genes. So I'm a bit curious of the performance of BC2 or Bandala in terms of fiber quality. Inosa is recommended for Visayas and Tangongon is recommended for Mindanao. The performance, the transcriptome performance of Inosa says that there are around 1/3 of its transcriptome upregulated. There are 2,065 genes that are upregulated, and there are 4,232 downregulated genes. While that of Tangongon, around 50% of the transcriptome population is expressed or upregulated. We also generated Venn diagrams across all the differential expression between Abaca and Pacol. And the results show that there are 733 commonly upregulated genes across all Abaca varieties. So we overlap each of the result of the differential expression. Like for example, here, Abuab versus Pacol, and then also BC2 versus Pacol, BC3 versus Pacol. So this is a pairwise differential expression between a high fiber quality genotype against Pacol, which has low fiber quality. So anyway, so the results show that there are 738 that are commonly upregulated across all Abaca varieties using this statistical and biological threshold. The results showed using gene ontology enrichment analysis of the PDE model indicates that glucan biosynthetic process, beta-glucan metabolic process, and 1,3-beta-D-glucan synthase and cellulose synthase activities are significantly enriched. And the genes that are associated with these terms include putative cellulose synthase and callose synthase. So these are potential candidate genes for further studies. So these are interesting genes. KEGG pathway analysis further says that shikimate O-hydroxynamol transferase, this gene, a key enzyme in lignin biosynthesis, is also enriched for this particular model. So again, this is another interesting gene. Genotypic differential expression model, which is a complex model, says that beta-glucosidase activity is significantly enriched. This model includes the comparison between Abaca varieties against Pacol. And the gene that is significantly enriched or are included in this term is glucan endo 1,3-beta-glucosidases. And again, these are the genes that are included in this term. We also implemented non-differential expression model, which is another complex model. This excludes Pacol, thus dropping the need to identify orthologous transcripts and the need to find differential expression genes between the two contrasting genotypic groups. So this involves the exclusion of the Pacol. So we only include only the Abaca varieties. So this is non-differential expression model. The results show that there are 3,737 of the 20,787 genes, which is around 18%, which are significantly expressed across the 5 abaca varieties using this statistical threshold. NDE gene ontology analysis showed a significant enrichment of beta-glucosidase activity and siloglucan-siloglucosyl transferase activity. Included in these terms are cellulose synthase like proteins and also putative cellulose synthase acatalytic subunit. Again, these are potential candidate genes. Gene ontology enrichment analysis revealed 7 different transcript isoforms encoding for putative siloglucan endoglucosylase hydrolase, protein 28, associated with siloglucan glycosyl transferase. Anyway, so this shows us that xylose and glucose and glucans are involved during fiber synthesis. KEGG pathway analysis also showed the significant enrichment of capiol cyclimate esterase and for fumarate CoA ligase, which are involved in lignin biosynthetic pathway. We also performed genotypic differential expression between susceptible and resistance. The susceptible varieties include abuab, inosa, and tangongon, and the resistant varieties include the back process and backball. So the resistance of the back process is conferred by backball. So anyway, so we performed differential expression using complex model between these groups. And the results show that 3 unique transcript orthologs which participate in disease resistance were found upregulated in the resistant group. And this include pathogenesis-related protein, PR1, and also Nibbler or non-binding site leucine rich repeats for disease resistance, and putative leaf rust 10 disease resistance locus receptor-like protein kinase. So these are interesting genes for further deployment for disease resistance or bunch-top virus resistance. Gene ontology enrichment of the genes that are upregulated in the resistant varieties include raw GDP dissociation inhibitor activity. In this term, this gene is significantly enriched, which is involved in phagocytosis. KEGG pathway analysis of the upregulated genes in the resistant group showed enzymes enriched in plant pathogen interactions. And this includes cyclic nucleotide gated channel, calcium-dependent protein kinase, and 3-ketoacyl-CoA synthase. So any recommendations? Yes, these genotypes were not infected, so we recommend challenging these genotypes with a virus. Such approach will elicit transcriptional response of genes associated with bunch-top virus resistance. So in summary, we have identified the genes that are associated with high fiber quality, such as cellulose, callose, lignin biosynthesis, xylose, glucan, etc., or these genes that are involved in the synthesis of such sugar or fiber. Pathway analysis showed the involvement of genes linked with lignin biosynthetic pathway for fiber synthesis, and candidate genes linked to bunch-top virus resistance in abaca were also identified, albeit modestly. BC3 and a backcross between abaca and wild banana was found to induce high levels of abaca-specific transcripts comparable to that of its parent, abua. So that's it. Thank you very much, and I'm ready for Q&A. All right, thank you very much. That's rather too short.
Thank you, Dr. Edeful. We hope that in the future, all the research effort you have done and your team has done would be able to eradicate this disease and bring about a positive change in the livelihoods and conditions of our Filipino farmers. For our next guest speaker, we would like to introduce Dr. Piwi Angeles from the Philippine Genome Center Program for Agriculture, Forestry, and Fisheries from UP Los Baños. Dr. Angeles received his PhD in molecular biology with a minor in bioinformatics from the New Mexico State University in Las Cruces, New Mexico. His master's in science in molecular biology and biotechnology minor in biochemistry is also from UP Los Baños. And his undergrad is from the University of the Philippines, Los Baños in BS Biology. He's currently a university researcher at the Philippine Genome Center Program for Agriculture, Forestry, and Fisheries, Office of the Vice Chancellor for Research and Extension, UPLB. Let us all listen to his talk titled Nurturing Epigenetics Through PGC Agriculture. Take it away, Sir Piwi.
Good day. Thank you again for the invitation to present our efforts on epigenetics at the Philippine Genome Center Agriculture at UPLB at this year's Genomics by Bioinformatics Conference at PGC. This talk is divided into two main sections. The first is to describe what epigenetics is all about and the various molecular mechanisms under it. The latter part would describe our work on epigenetics at PGC Agri at UP Los Baños. Epigenetics is described as heritable states of gene expression that are not dependent on changes on the DNA sequence. These changes are highly dynamic throughout the life of an organism, both spatially and/or temporally. The field of epigenetics encompasses 5 major molecular mechanisms. The first is the modifications of nucleic acids, both DNA and RNA, which is exemplified chiefly by 5-methylcytosine. Related to that is the mechanism of genome imprinting, wherein depending on where the methylation would lie that one gets from the parent would result to altered transcription rates. So for instance, in this cartoon, methylation at this part of the paternal strand turns off the transcription of gene A but activates gene B. In contrast, if you unmethylate now the maternal strand, you activate the expression of gene A, but now repress gene B. Another molecular mechanism under epigenetics involves the transcription, activation, or repression as a result of the differential chemical modifications on the amino acids protruding from the core where the DNA wraps around. So depending on what type of chemical modification is on these amino acids and where these amino acids lie in these protruding parts of the histone core, you'll get either transcription repression or transcription activation. Related to the histone code modifications is how these chemical modifications on the protruding amino acids would allow either further attraction of adjacent nucleosomes or further repulsion of the nucleosomes to effect either transcription repression if you allow these nucleosomes to be more associated to each other, or have a further repulsion. These DNA sequences would be opened up and would now allow transcription machinery to come in, allowing transcription activation. The last molecular mechanism under epigenetics is the activity of non-coding RNA, both the short non-coding RNA, the siRNAs and the microRNAs, for instance, as well as the long non-coding RNAs or linked RNAs, typified by EXIST, which is the one of the chief initiators of random X chromosome inactivation that is found that is exemplified by the calico cat. All of these different molecular mechanisms lead to altered gene expression, resulting in changes in the phenotype without the change in the DNA sequence. These epigenetic processes are found both in prokaryotic and eukaryotic systems. And in eukaryotes, we find these in plants and in animals. In plants, for instance, epigenetics are involved in changes in the phenotype, in reproduction and the development of an organism, in the ability of an organism to respond to stress, either internal or external to it. And in some cases, like DNA methylation, it can be, these methylation marks may be inherited in subsequent generations. These epigenetic phenotypes may also be found in animals, and in addition to the ones described, other animal-specific phenotypes include aging, cancer, the development and function of the brain, behavioral epigenetics, for instance, how an offspring would behave in relation to the behavior of its parent to it. Nutritional epigenetics involves how the nutrition that we get from our environment affects us or have a phenotypic effect on the organism. And lately in 2010, methylomes or methylation patterns have been used to describe phylogenetic work as well. Over at PGC Agriculture, epigenetics is one of the 5 functional groups that we use to for our stakeholders, and this functional group is headed by our program director, Dr. Tony Lorena, with yours truly as the assistant leader. We apply epigenetics towards understanding our different commodities and stakeholders, including crops, livestock animals, marine and forest organisms, microorganisms, and arthropods. We've been involved with several aspects on epigenetics for research, instruction, and extension. And we have an ongoing project on this, which with yours truly as the program leader. And I also had the studies on research and instruction with one of my colleagues, Jeric Villanueva, handling the extension part of the work. Our first foray into epigenetics research is to assist Dr. Dennis Belaong in analyzing his work on regulated microRNAs in teleost fishes. This work was presented at the Philippine Society for Microbiology in 2016, and with further computational analysis, we packaged that as a poster and presented that at FAO-BMB, PSBMB later that year. We also moved into DNA methylation research, and we've put out a couple of publications related to that, in particular to how epigenetics or DNA methylation in particular could play in our different important coconut endosperm phenotypes. So we put out two publications. The first is in the special edition of Agronomy for Plant Epigenetic Mechanisms, together with Dr. Christy Cueto at the Philippine Coconut Authority, and some members of the Genetics and Molecular Biology Division at the Institute of Biological Sciences, UP Los Baños. We further expanded that text. We added more to it and more discussions. And this was contributed as an invited book chapter in the recently published book entitled The Coconut Genome, which got out in September of 2021. Throughout the years, we've also engaged in developing research proposals on the field, ranging from basic, including a proposal on understanding how promoters could be used to produce pharmaceutically and industrially important proteins. But much of the proposals so far are on agriculture-related subjects, including how methylation could be used to study how crops adapt to nutrient stress, how the same crop could be affected by positive biotic supplementation, or when you add something that will help it grow further. Several units at the university has also engaged with PGC Agri in other omics-related work, including epigenetics, on how organisms and its byproducts from our endemic flora and fauna may be used for clinical applications when tested on model organisms. The last proposal that we've done is on coconut epigenetics, which stems from our published work that I presented a while ago. We've also ventured into a proposal on fisheries and aquaculture and try to understand how epigenetics and the other omics fields play a role towards the environmental adaptation of one of our salient endemic fauna. This proposal, amongst all the proposals that we've done, has garnered much traction over the past couple of months, and we hope that further positive development will arise from this proposal in the next few months. We've also engaged the community in instruction-related activities for epigenetics through the Institute of Biology, UP Diliman, and we offered a graduate-level special topics course on epigenetics. We first offered that in 2018 with 4 enrollees. There were no enrollees in 2019, but when we offered that again in 2020, we had 9 students. We hope that this upward trend in enrollees could be sustained when we offer that again come the second semester of 2021. We also engaged the community towards extension-related activities for epigenetics by putting out an online course in the field that we did in September 2020 all the way up to January 2021. Out of the 9 participants that we had, 3 completed the entire course and were awarded certificates of completion. We packaged that course along with Bio 397 into a summarized version on epigenetics, and we presented that as a webinar last May 2021 that was co-hosted by PSBMB Southern Luzon chapter. And in that webinar, we had 87 participants. This webinar is now offered as a webinar series, and we started that work last November 2021 with a webinar on the basic tenets on epigenetics, and we had 76 participants on that. In December of 2021, we plan to hold a webinar on plant epigenetics and conclude the webinar series in January of 2022 with a presentation on animal epigenetics. in summary, we at PGC Agri work and engage the community on epigenetics. So I presented what epigenetics is all about and the various molecular mechanisms under it, and gave you a snapshot of what we do on epigenetics at PGC Agri. So if you are interested with the field and you want to engage and network with us, please, we highly encourage you to reach out to us through either our organization contact details here or through my own personal contact details. Again, thank you for the invitation and have a great day.
Thank you, Dr. Peewee, for bringing attention to the application of epigenetics to crop enhancement, animal health, and aquaculture, where we can offer scientists and researchers a wide horizon of opportunity. It's worth educating the Filipino people on the opportunities of epigenetics as a tool to build resiliency in the face of a global pandemic and climate change. We're excited to hear more of your research, hopefully next year. Don't forget to sign up once it's available. For our last speaker today, we would like to welcome Ms. Lilian Pagaduan Villamor from the Philippine Carabao Center. Ms. Villamor is a senior science research specialist at the Cryobank unit of the Philippine Carabao Center. Her main responsibility is to enhance the operationalization of cryobank in areas of molecular identification, cryopreservation, database and information campaign. She plays a vital role in strengthening linkages to farmers, researchers in the government agencies, private sectors, academia, and other stakeholders in order to maximize the utility of the cryobank facility for in vitro cryoconservation of livestock and related species, as well as indigenous wild, threatened, and endangered animal genetic traces. She has gained skills in the management and operations of the cryobank through international collaborations with Korea, through the Korean International Cooperation, and through the Korean Animal Product Evaluation in Taiwan. And she has also collaborated with the National Center for Genetic Resources Preservation of the USDA. She obtained her bachelor's degree in biology in 2002 from the Brigham Young University, Hawaii, through an International Work Experience Scholarship Program. She has earned her Master's in Science in Marine Science with specialization in marine biotechnology from the University of the Philippines, Diliman. She is currently pursuing her doctorate in bioscience through research from the Tokyo University of Agriculture, Japan, to the Tokyo Nodai Sirka Scholarship for dissertation doctoral degree. Let us all welcome Ma'am Lilian for her talk, Water Buffalo Research in the New Normal, the PCC Experience. You can proceed, ma'am, and share your screen.
Okay. Okay, am I loud and clear to everyone?
Yes, ma'am.
Okay. Sige, magandang hapon po sa ating lahat. Bumabati po ako ng magandang hapon mula sa Kalabawan dito sa Nueva Ecija. I'm grateful that Dr. Yap and the organizing committee invited me to be one of the resource speakers for the 2021 Genomics and Bioinformatics webinar to share with you the challenges and the opportunities that were experienced by the Philippine Carabao Center during the COVID-19 pandemic. I entitled my presentation, Water Buffalo Research, the New Normal, the PCC Experience. If you haven't been to the Philippine Carabao Center, it is located in Nueva Ecija, and it will take a 3 to 4 hour drive, considering no traffic, to reach our national headquarters. And I am working under the Research and Development Division in the Cryobank unit, which I was able to manage the ex situ cryoconservation and molecular characteristics of animal genetic resources in the cryobank, which has been inaugurated last September as the National Livestock Cryobank. The Philippine Carabao Center is an attached agency of the Department of Agriculture that is mandated to conserve, propagate, promote the carabao as a source of milk, meat, draft, and hide to benefit the rural farmers. Water buffaloes are actually of two types, the riverine, which produce meat and milk, and the swamp buffaloes, which are source of draft power in cultivating farmlands. It's inevitable that pandemic extremely affected the carabao industry, from employment in different carabao cooperatives, services to animals, forage supply, or which we know the food supply for the animals, the veterinary supplies. And this boiled down to result in the low distribution, processing, and marketing of milk. And actually, based on the survey and the data, 22% decrease in milk production was recorded in these during the pandemic. Pandemic truly slowed down the work in carabaos, especially milking these animals. But the farmers continued to do the work. So tuloy-tuloy ang paggatas, so tuloy-tuloy din ang pagtatrabaho, especially for our the frontliners, the buffalo farmers who work hard even though there is pandemic. And the pandemic allowed the creation of the program known as Alpas contra COVID, which means Alpas in Tagalog means kumawala, or in English, to become free or break loose from the COVID-19 pandemic. This presented 4 strategies. The first one is the ULAP, which stands for the Unlad Lahi Project. The second is on the COVID, which means creating opportunities through value innovations and development. Third strategy involves the GGKs, that stands for Gatas, Gulay at Karne, and the fourth one is Kara Aralan. Kara is the abbreviated word for Carabao. And let me walk you through these different projects that our agency done, especially during the pandemic, when we had lockdowns in our country. So the first program is for the Unlad Lahi Project. It is a provision for animals, incentives on technical and veterinary services to displaced farmers and carabao owners. Incentives to Vivaites. The Vivaites are known as the village-based and artificial insemination technician. And the objective of this project is to increase milk and meat production, job creation, and providing livelihood for displaced farmers. And one of the outputs is the establishment of the ruminants clinic that is open not only in provinces of Nueva Ecija, but across the Philippines. Even though there is lockdown, the Vivaites were unstoppable. With proper use of PPE and following the health protocols, they continued to extend services for artificial insemination and assisted health concerns of the animals. Ito po yung mga farmers natin sa Pilipinas na gumigising ng maaga para po matugunan ang mga pangangailangan ng ating mga kalabawan sa Pilipinas. And the second strategy is the COVID, which imply the rolling of technologies and innovations of food production, processing, and value adding. And this has the objectives to enhance the food productivity, availability, accessibility, and sustainability through applicable technologies and valuable innovations. And this also created livelihood to our farmers. One of the outputs of this project was the development of Milky Bun and milk supplements. So these numbers that you can see in here is the total number back in 2020, 78,000 distributed to various individuals for the students, for the feeding program, and also utilized for ayuda for the frontliners. So ang tanong, bakit gatas ng kalabaw? Ang sagot doon, bakit hindi? Since the carabaw's milk is a nutritious source of calcium, calorie, potassium, phosphorus, and magnesium, this is really a good source of immunity booster. And when we were dealing with the COVID-19 pandemic. So even though there is pandemic, there were technologies that were developed, and this is few of those technologies that were developed by our research and development division headed by Dr. Eufrosina Atabay. And these technologies were done for the improvement for the health, full supply, and production of carabao. Moving to the third strategy of this ALPA's COVID, contra COVID, this is the concept is like the farm to market strategy. So there were clustered vegetables and livestock, forage production, market assistance. So basically the farmers brought their produce, their products in here. We had it in Milkakram, which is in front of the national headquarters. And basically this attracted the local clients to appreciate their produce and buy them with reasonable amount. And another effort that was done by the nutrition department, actually the nutrition unit in our RDD, is the development of the Bura Booster, which is a silage inoculant, and it serves as an additive for corn, sorghum, and grass silage. And this increased the food supply for the milking carabaos. Another innovation under the COVID strategy is the BMW, known as the Buffalo Milk on Wheels. Actually, the story behind this BMW, it's originally a mobile laboratory previously used for addressing the animal's health and production. But since the pandemic hit the country, this was transformed into a delivery van for Buffalo dairy products. So everyone was involved, whether you are in a top management, researchers, and even scientists, they were involved in marketing. Actually, this picture that I showed on the right side is one of the scientists, Dr. Dunilda Duran, who was spearheading the buffalo on wings in some provinces in Nueva Ecija. And the good news, the BMW rolls out in selected towns, cities across the Department of Agriculture PCC network in Luzon, Visayas, and Mindanao. In our team, the Cariobank team, we found a comfort on doing online marketing. We contacted our friends, our previous employees, and interested clients via social media to be online resellers of the Carabao dairy products. And we refer the interested clients to our marketing coordinators, and this worked well to strengthen the dairy business on Carabao dairy products during the pandemic. And as a matter of fact, within 3 months, from March to June 2020, 92 million liters of milk were processed that were utilized for ayuda, for frontliners, as well as for milk breeding and also for marketing. And I just want to give credit to these online resellers from 15 areas in North and Central Luzon that provided 1.3 million worth of milk that they were able to sell through online marketing. And the pictures that you see right now are the top 5 online resellers across in Northern and Central Luzon. And actually, 2 of those are actually students during that time, and they're earning money through online reselling, in the same time helping the farmers to sell their products. And for the fourth strategy is the Kara Aralan. And this brought the e-learning webinars focus on farm solutions. And you can see how our farmers be able to adapt and learn to use the technology in order to gain knowledge through these e-learning webinars and modules in video farming. So basically, what are the lessons learned during the pandemic? There are 2 points. One is the Alpas contra COVID provided strategies to help farmers rise above the challenges of the pandemic and adapt to the new normal. Pandemic inevitably slowed down the productivity in doing research, but it brought opportunities to create, innovate, and improve the way to address the needs of Carabao farmers and carapreneurs. How about the basic research? Prior to pandemic, we have involved in working on genetic diversity and species identification of the Philippine Carabao, which has implications to conservation and management. And part of it was a project funded by the Department of Agriculture Livestock Biotech Program. And this is entitled Genetic Diversity of the Philippine Carabao using the Mitochondrial DNA CO1 and Microsatellite Markers. And this is to recognize the project staff, Dr. Esther Flores, Dr. Sarabia, and the researchers, Mr. Paraguas, Ms. Exquadro, and Ms. Kailipan for doing this work. And the main objective of this study was to determine if there is really a genetic diversity in the Philippine Carabao. This simple yet pertinent question to be answered by the study would imply 2 possible directions toward the in situ conservation of the species. First, if the answer is no, meaning there is no genetic diversity of the Philippine Carabao, this entails that the present institutional conservation of Philippine Carabao at the Cagayan State University will be sufficient to represent the genetic traits of the Philippine Carabao in the country. But if the answer is yes, meaning there is genetic diversity of the Philippine Carabao subpopulations, the genetic diversity of the Philippine Carabao will refine the rationale of strengthening the current, but not limited to expand the strategic conservation and management of the Philippine Carabao in the country. Sampling sites were considered from isolated areas across the islands of Luzon, Visayas, and Mindanao with a total of 27 subpopulations. And this is just to show you the landscape of the sampling sites. We walked, we hiked, we crossed the rivers, the seas, and but this sampling was manageable before the pandemic with proper coordination with the LGUs and PCC centers. And it is all worth it to be with the farmers who are very hardworking Filipinos who are dedicated in taking care of the animals, as well as cultivating the farmlands. And they are indeed source of inspiration being with them. And this is a picture of one of the sampling sites in northern Luzon, specifically in Calayan Island, wherein our project contributed to identify this island as a Carabao sanctuary in Luzon. So going back to the basic research on our project, this is basically the methodology of what we have done, and you're basically familiar with it. From blood collection, we isolated DNA, did the PCR amplification and fragment analysis, as well as sequencing and phylogenetic analysis. But I wanted to point out on the experience that we had for blood collection. We understand that DNA is very vital for the downstream analysis for our different projects. But due to some cancellation and logistics in the LGUs, we were not able to do additional sampling for these animals. And the big question was, what would be the source of biological samples for DNA extraction? So basically, with our initial project on genetic diversity, we didn't have much because we also have other studies that we performed. But it's a good thing that there is a National Livestock Cryobank, which is fortunately housed in the Philippine Carabao Center. And this served as the repository of genetic resources. So just to share a glimpse of the cryobanking in the Philippines, this is an ex situ cryopreservation for animal genetic conservation, leading to sustainable animal production systems. And you will see these 6 huge tanks, cryotanks that serve as repository for long-term storage of frozen semen and oocytes, embryos in liquid nitrogen for future utilization, specifically for artificial insemination in various cooperatives and local government units, as well as for other research for in vitro fertilization. There are 2 types actually of biological samples that are being maintained in the cryobank. The first one is the reproductive samples, which I mentioned previously, the semen, the oocytes, and the embryos. And they are all have to be preserved properly so that they will still be viable when being used for production of animals or introduced in the population through artificial insemination. The second type are those non-reproductive biological samples, which include the DNA tissues and the cryopreserved blood. Going back to before the pandemic, our strategy before the pandemic was to back up the DNA and the cryopreserved blood. Our foresight during that time was to preserve the blood as an additional backup to save time and money from resampling animals for other studies. True enough, cryopreserved blood provided a source of DNA to finish our project and pursue other species and genetic diversity studies during the pandemic. My message for those engaged in molecular analysis and studies is to plan to preserve your genetic resources for future utilization. And for those who are interested to back up their samples, the cryobank, the National Livestock Cryobank in our unit are willing to help you out for that purpose. So we're happy that we are able to obtain results and completed our study on the genetic diversity of the Philippine Carabao, and that going back to our appreciation to the cryopreserved blood for sources of DNA. So to share with you some of our results, this is the landscape of the genetic, the population structure of the 77 subpopulations of swamp buffaloes across Luzon, Visayas, and Mindanao. At optimal K value of 3, there are 2 swamp buffalo clusters that were identified. Majority of the populations in Luzon, shown by this blue vertical color, was identified as 1 swamp buffalo cluster. And if you will notice that the southern part population in Luzon, from Carabao, Sorsogon, Occidental Mindoro, showed affinity with those clusters in Visayas and Mindanao, represented by the green vertical component. And the third cluster is identified in the riverine component, riverine riverine individuals with affinity of these individuals with represented by the red vertical component. So to focus on the population structure in selected Luzon population, this show that Ilocos population has an admixture of riverine and swamp buffaloes. However, if you will focus on the Calayan Island, this indicated pure swamp type with membership coefficient of 96 to 99.6%. And this confirmed that the animals or the swamp buffaloes here in Calayan are worth of conserving because they're still undiluted. And to explain the relationship of the southern Luzon population Southern Luzon population with the Visayas and Mindanao, this revealed 3 points: that Carabao Island, Sorsogon, and Occidental Mindoro in Southern Luzon showed different cluster from Northern and Central Luzon. But it showed closer affinity to Visayas and Mindanao. And as you notice, that Visayas and Mindanao are shared common ancestry than Luzon populations. And the highlight of this study also revealed that the Swamp 2 cluster were identified in populations in Carabao Island in Romblon, Gigantes Island in Iloilo, and Pitogo Island in Bohol. So after determining that there is indeed genetic diversity of the Philippine carabao, what is now the implication to the in situ conservation management of the Department of Agriculture? The basic information that were gathered here from the genetic diversity of Philippine carabao provides useful insights for policy and guidelines on a strategic conservation and management program. And for the implication to ex situ conservation and management, it is worth it to store the germplasms from Calayan Island and Pitogo Island in Bohol as sources of genetic resources for Swamp Cluster 1 and Swamp Cluster 2. And for this next study, it highlighted the genetic diversity of the Philippine carabao using the mitochondrial DNA dilute variation and implication for conservation as well. I just want to recognize the contribution and the mentoring provided by Dr. Konomura, Dr. Takashi, Dr. Amano from the Tokyo University of Agriculture and Yamazaki University of Animal Health Technology, and the funding agency from CIRCA. And this provided us a better understanding of the genetic diversity on the mitochondrial DNA for the swamp buffaloes. Just to give you a little bit of background of the domestication of the swamp buffaloes, this started in China region, that after the domesticated Chinese swamp buffaloes, it revealed 2 dispersal pathways. The first one is through Taiwan and the Philippines to the eastern islands of Borneo and Sulawesi. And the second pathway was through the mainland Southeast Asia and then to the western islands of Indonesia. Genetic diversity of Asian swamp buffaloes reported that there are 2 lineages of swamp buffaloes in Asia. These were reported based on the cytochrome B and mitochondrial DNA, that the predominant swamp lineage A is very predominant in the Asian countries. However, in the Philippines, we have limited study on the genetic diversity. One of the reports that we were able to contribute was to identify these swamp buffaloes in Calayan Island that helped to determine that these swamp buffaloes belong to that are worth of conserving because they are pure type swamp buffaloes. Previous study on the genetic diversity of the swamp buffaloes in the Philippines reported by Del Barrio et al. That only swamp lineage A was detected in the Philippines based on their sampling collections in Philippine carabao centers across the Philippines as well as in organized cooperatives. This was confirmed by another study by Yu et al. In the phylogeography and domestication of Chinese swamp buffalo, that Philippines and other buffalo sequences outside China were only incorporated in lineage A and no sequences fell into lineage B. With our study, with the comprehensive study of this genetic diversity of swamp buffaloes, we determined or detected that there are still 2 lineages present in the Philippines, the lineage A and the lineage B. For the previous study, we confirmed that lineage E is still the predominant haplotypes in the Philippines, and 2 haplotypes were only detected in lineage B from Occidental Mindoro and Capiz. So the pattern here demonstrated that a closer affinity between few haplotypes are those with geographically close populations and had improved accessibility to transportations, as manifested here in haplotypes H13 for Capiz, Gigantes Island between the Visayas and Mindanao populations, and also in the North and Central Luzon in haplotypes 4, 12, 8, and 19, and 9, I should say. The median network of 107 Philippine Carabao samples determined the prominent haplotype in lineage A that has a pattern of decreasing haplotypes from Luzon, Visayas, and Mindanao. And this is also considered as the ancestral haplotype. And the second prominent haplotype was detected in H2, which showed a pattern of increasing, which is opposite in the haplotype 1, but this haplotype has an increasing haplotype diversity and nucleotide diversity from Luzon to Visayas and Mindanao. And this pattern of haplotype distribution demonstrated the existence of an evolutionary relationship among these populations. This is simply showing the distribution of the frequency of the 16 haplotypes in the Philippines. This mainly would like to emphasize that there is indeed genetic diversity of the swamp buffaloes in our country. And most of the haplotype diversity and nucleotide diversity were high in Calayan Island, Capiz, as well as in Bukidnon. Having said about this genetic diversity that we are able to detect in our study, this has a great implication in the conservation and management of the swamp buffaloes in the country. Currently, the gene pools in Cagayan State University, in Calayan Island, in Carabao, in CPG, Native Carabao in Bohol, these are 3 in situ conservations, and the in situ conservation, as well as the ex situ here in the national headquarters. These conservation efforts were able to secure the swamp genetic resources that are limited to haplotypes belong to lineage A. Thus, this study recommended that swamp genetic resources from lineage B would be recommended for strategic conservation and management program. And what do we need to back up for our ex situ conservation? We are considering collecting, storing, and preserving the germplasm from genetic resources of lineage A from Calayan Island and also in Bukidnon, and also genetic resources from lineage B from populations in Occidental Mindoro and Capiz. So in summary, the presence of genetic diversity and population structure of swamp buffaloes were identified across Luzon, Visayas, and Mindanao. Distinct clusters of pure type swamp were detected in swamp cluster from Calayan Island, Cagayan, and swamp cluster 2 in Carabao Island in Romblon, Gigantes Island in Iloilo, and Pitogo Island in Bohol. The mtDNA developed sequences of swamp buffalo across the Philippines demonstrated the major and minor groups identified into maternal lineages A and B respectively. And moving forward with this genetic diversity, we are collaborating with the PGC as well as with the USDA for the whole genome sequencing of the swamp buffalo genetic resources. With that, I would like to acknowledge the funding agencies as well as the farmers from different regional centers and the LGs and the VIVAID and the DA regional staff for contributing to these studies and also other efforts of the Philippine Carabao Center. And again, maraming salamat po. Thank you very much.
Maraming salamat, Ma'am Lilian. With that talk, I think the participants would like a short break to drink milk. So let's get back into our conference after 5 minutes.
All right. So good afternoon, everyone. Okay. Good afternoon, everyone. Okay. Welcome back. Hope you've gotten the chance to freshen up. Okay. So first of all, there's nothing wrong with your screen. I'd be John Michael Yap, the Program Director of the Core Facility for Bioinformatics at the UP Philippine Genome Center, and I'll be taking over from Miss Shalette Monzon, who's done a stand-up job hosting the first parts of this day's festivities for our GBCon 2021. Okay, I'll be handling the open forum and the latter parts of this program. Okay, so before we truly begin with the Q&A portion of today's events, okay, I would like to request the speakers, Ma'am Heidi, Sir Peewee, Sir Nelzo, and Ma'am Lillian to kindly turn on your video, okay, so that the spotlight can be put on all of you, and we can proceed with the Q&A. Okay. Now, for our audience, okay, for the participants, you can still write in or put in your questions. Okay. While the current while the the asking and the answering are going on. Okay. So that if you have something on your mind that you wish to manifest or to ask our esteemed guests, esteemed resource speakers, you could just we could try and accommodate that. Okay. So here we go. All right. So good afternoon, everyone. Okay. Actually, the first question that I want to ask is more of an extension of already answered questions, actually privately answered questions, okay, by some of our participants here. Okay, so I would like just to extend, since the theme of this conference is overcoming challenges and building opportunities, right? Could you provide or give details on opportunities that you have within your respective spheres of discipline or units, okay, for both budding scientists, be it students or young scientists, or even senior researchers, on what they could do together with your institution, together with you, okay, towards developing your research interests. Okay, so let's start first with Ma'am Heidi.
Hello, good afternoon again. I guess I mentioned some of those already as I walked through beyond coconut, how overall, because coconut was not that affected in conduct, since we more or less we have finished towards the end of 2019, and some more of the admin and the wrapping up of the projects. But for the other ongoing projects, I guess it's the resilience of the Filipino in general, and of course we are so more or less, at least at our experience at UP, we really able to kick back immediately to address both research and instruction and extension and all related academic assignments. So in response to for students, of course, there are ongoing research. This is students that time in the middle. Those that have started have to really find. Bioinformatics came handy when you have already data, and just try to really do because we have the we had the hard lockdown, but they were opening and just 30%, 20%, 10%. There was even 10%. But it was very important if you are clear what are the critical of your ongoing research experiments. And in terms of prioritization, of course, I guess in priority are those that are in critical of there is ongoing research because we restricted a number of people to work in the lab or any workspace. So it's really the giving and sharing. And then for the students, especially those graduating or already in their last terminal to already complete, finish, when there are scholarship that will expire, then they are one of the first priority to support. So it's all support. And then since administration matters also affect everyone, instead I mean, the experience is really like, instead of really finding it as the problem, but meet up and offer solution, because everyone was on the situation of from hard data documents and then direct just immediately migrate to online submission. But yet still we know that in accounting system and some auditing rules, we still have to submit hard documents. So all in all, we manage and we arose from the challenge, still under situation now. It's so challenging, but all those publications, data analysis, and strategic planning were just the time that were maximally used to lay ground the foundation and the planning during those hard lockdowns.
Okay.
So direction setting.
Direction setting. Okay. Thank you very much, Ma'am Heidi, for that. How about you, Sir Nelzo? Opportunities from your neck of the woods.
Okay, opportunities. I have one opportunity for a PhD or MSc student. If you were asking, can I share my screen?
Yes, you
I
may.
think it's,
Yes, yes.
I think
If
it's
only
done.
for a few seconds.
Okay.
Understand.
Only for a few seconds. Okay. So okay. So this has been recently funded, but yeah, so we're looking for a MSc PhD student to undertake regulatory divergence analysis and heterotic variability between banana. So if you are that person, you have molecular biology, ag biotech, or agronomy, and with these desirable skills, if you're interested to pursue that one, kindly email me. Okay, so that's it. That's very quick, and sorry for being too specific, but I think that's the best answer that I could give you right now.
Actually, Sir Nelzo, that was perfect. That was perfect. Thank you very much for that. That's actually, something that it could easily be, processed and grabbed by interested parties in our audience. Okay. Now, next, Sir Piwi, how about you? Some opportunities from the epigenetics and PGC agriculture side.
Right. So I'll start off with education probably. So we have the epigenetics graduate course through UP Diliman in the second sem. So if you're interested to pursue epigenetics, like the theoretical side as well as the application side, you can do that. Extension side, we have ongoing webinars. We have one for next week on plant epigenetics, and then another one on January 22 for animal epigenetics. Plus, of course, other things that we can work on. You can also engage with PGC Agriculture, as for example, for epigenetics. If you want to do that sort of work, there's only about less than 20 scientists and people who do epigenetics in the entire Philippines. And those are scattered all throughout the 5 different epigenetic molecular mechanisms I discussed. I'll do research last. As a footnote, we just obtained permission or a start of implementation notice for epigenetics and other omics fields to understand the environmental adaptation of one of our salient fishes. And so that will open up in the next few weeks, probably. So research opportunities as a student and as staff members for those. So that will be for genomics, transcriptomics, and epigenomics. It's coming out in the next few weeks.
Nice. Thank you very much, Sir Piwi, for that. Lots of opportunities coming in, even in the near future. Now, how about you, Ma'am Lilian? Opportunities coming from the Philippine Carabao Center.
Okay, we understand that our research is still anchored on the ALPAS contra COVID. So basically those, if you like to work with farmers, Buffalo farmers, you are so welcome to join our institution, even to do research. So we have a wide spectrum of research in the Department of Agriculture, Philippine Carabao Center, that would need a contribution on improving milk, for breeding program, for conservation. And now where I'm at, here in the cryobank, as a National Livestock Cryobank, we are inviting researchers to work on genetic resources for identifying which species, which species are worth of conserving, as represented as in the cryobank, that will preserve the genetic diversity of our wild, indigenous, and commercial breeds of animal genetic resources. So basically, we also have other units that are working on animal health. So basically, if you are in line with that, we are also promoting and inviting more researchers to work with our co-researchers. And if you are interested, please don't hesitate to message us, and I can link you to that, or even work with our own unit here in the cryobank. Thank you.
Thank you very much, Ma'am Lillian. Exciting opportunities about not only for COVID related, but also for animal science focused on Carabao, genetics, genomics, and even other how should I call it? Livestock science. Okay, cool. Okay, so okay, there's no one posting. Okay, I guess I could ask another question from my end. So I already touched upon the opportunities that each of your units or each of you have for our dear guests, right? Our dear audience members. Now, of course, they also have to understand that there are going to be challenges that have to be faced, okay, once they try to grab that opportunity. So can you give probably one or two major challenges, okay, that they will be facing should they join your respective teams? Okay, now I went counterclockwise, starting from Heidi, at least basing from my view here. I'll start again with Ma'am Lillian. So challenges. Once an opportunity was grabbed in your unit.
Okay. One of the challenges that we had is we are highly involved in sampling collections. So with that, with many lockdowns and also different IATF protocols to adhere, basically our sampling collections were hampered, and genetic resources in our biotech research have been hampered as well. So basically, what saved us to continue doing our research are those deposited genetic resources that we have. We have the semen samples and oocytes, and mainly like what I discussed earlier with my presentation is that the importance of securing the DNA, also the other biological sample tissues and cryopreserved blood that will be sources of DNA for downstream analysis. So with that, because of that, we were able to do a lot of research on the genetic diversity, species identification, and serve as reference samples to other research in our agency.
Thank you very much, Ma'am Lillian. At least they're now aware of the challenges that they also have to face once they try to join in the fray of in PCC's work. Okay. Now, Sir Peewee, how about yours? How about challenges?
Challenges. I'll discount already our administrative and operational challenges. So I'll just focus on the field of epigenetics in the Philippines. Again, there's only less than 20 probably people who are doing epigenetic research. So it's a very, very young field in the Philippines. But if you look at it in the global perspective, that's already like built in, especially if you want to do transcriptomic research. But so the mere promulgation of the field in the Philippines is a big challenge for us, I think. So that's why we hold seminars and webinars and promulgate it through this fora. And of course, by doing so, we want also to engage with the community on the possibility and the applications of epigenetics, not only for eukaryotes, but also for prokaryotes, which we haven't really done yet. I haven't seen anything yet on the prokaryotic side. So, there's still a lot, much work to do on research, education, and extension work on epigenetics, I think.
So in a sense, it's mainstreamification or popularization
Right.
of epigenetics. Well,
Compared to maybe like genomics or transcriptomics, that is
Ah.
like widely more,
Yeah.
known,
mhm.
but epigenetics, not much.
Well, on one end, that can also be an advantage. It's a young field, right? A lot of things that can be done in a sense here and locally. So at least that also can be turned into an opportunity in a sense. That's
Yes,
that's nice. That's
yeah.
nice. All right. Thank you very much, Sir Peewee. Sir Nelzo, how about you? Challenges.
Right, okay. I'm not sure, John, whether you could read the questions on the Q&A that were sent privately, but I can read the question, and then if you are not
Yeah.
able to respond.
Yeah, sure.
Okay.
Actually, I could. Yeah.
Anyway, so answering your question, well, first, the current pandemic, our current situation is posing challenges to us. That's one, which is obviously being experienced by everyone. Anyway, the second one is the shortage of bioinformatics facilities on our side, at least in the PGC UPLB. I think that's one of the challenges right now, because we are moving towards this discipline, and so I think that we have to invest in this kind of facilities or in this discipline. And thirdly, if you are starting to learn computational approaches, then learning it is a challenging curve. I mean, I'm not sure how emphasized or advanced, because I'm relatively new in the Philippines, how advanced our bioinformatics. To address partially those things, we have conducted bioinformatics workshop recently, like a couple of weeks ago on RNA-seq to start addressing this kind of issues. So we have conducted RNA-seq analysis from pre-processing to differential expression, and there might be some coming workshops in the coming months, like variant calling and then 3D RNA-seq. So we have to develop this discipline because it's relatively young in the Philippines. But again, I'm not sure how really it is, how young or how old, how advanced it is in the Philippines. Yeah, I think that's it.
Okay, thank you very much, Sir Nel. So computational capacity building, both on the equipment side and also on the expertise side, human resources side. Definitely equipment, we need that. Computing power is never enough. As for the human resource capacity, as far as I know, there have been special topics or specialized courses being offered by several units around the Philippines, not only UP. I could name, for example, Mapua and De La Salle University as those, and even MSU IIT, in fact, right? But I think it's more of a course, rather than a degree program. So I guess it may also be it's kind of a cold shot right now that to let you know, sir, and also those in the audience and those watching, that the UP, the University of the Philippines, is trying to prepare a bioinformatics graduate programs actually, and they're going through now the process of instituting that. So lots of birthing pains at this point. Hopefully in the near future, probably next year or in the next 2 years, that will be established. But also there have been already a lot of courses, and there are a lot of even research groups that have been engaged in bioinformatics. I guess a unification of sorts, a unifying group, so much so that there's a network, so that there could be exchanges in terms of expertise, and probably even ideas could also help. Okay, so that's Sir Nelson. Thank you very much. Okay, Ma'am Heidi, how about you?
I guess I just add to that as an opportunity because of the pandemic, that initial fund prepared to lay down the foundation of the previous planned PGC Central Laboratory. Otherwise, if we have spent that, we don't have a much fund for the COICA project, because the foundation should still be for the beneficiary. So given that, and all things well, because of course it's still in the initial implementation. There are still many things to happen. That should be a good capacity, facility, and more academic and human resource building for bioinformatics and omics application. So that is what I'm presenting earlier. I didn't give detail because later on there will be with the PGC Agri team, but that's for the putting up of the UPLB Agricultural Genomics Center and house for facility, including bioinformatics and all other wet lab omics research. And we'll welcome also academic. There will also be the plan for a co-development of a curricular program for omics application, and of course come with bioinformatics. So that's very bright to look forward. I hope that will be inspiring for everyone, and network for all genomics and bioinformatics nationwide.
Fingers crossed. Yeah. All right. Okay. Thank you very much, everyone. Okay. We still have a minute. Okay. Maybe you could give a 20-second final message. Okay. This time I'll call by alphabetical order. I know surname. Okay. So we start with Sir Peewee. 20-second message to the audience.
Right. Thank you, Dr. Yap. Again, thank you for PGC for inviting us, PGC Agri, and the different omics. So I just want to share with you or leave an imparting message that genomics is not just genomics, it's a whole slew of omics that you can go into depending on your liking and depending on your interest. And I hope this could serve PGC, and the rest of the PGC centers can help in engaging with the community on these. Thank you.
Thank you very much, Sir Peewee. Next, Ma'am Heidi. Ma'am Heidi, 20-second final message.
Again, we are so grateful and thank you very much, PGC, for organizing this one and for inviting us. And it was also a nice time for us actually to collate and reflect what actually we have done in spite of, and realize that we are not actually really backlogged. I mean, like we are slowed, but things are brighter along the way. And as I said, one step at a time, small efforts will be a big success for everyone, helping together.
Thank you very much, Ma'am Heidi. Sir Nelzo, your message. Kindly unmute, sir.
Oh, sorry. Okay. Okay, 20 seconds na yun, ah. Anyways, I'm so grateful. Thank you very much for inviting me as one of the speakers for this workshop or for this seminar, series of seminars. And the only message I would think is that everyone has his or his skills in pushing the wheels of science, especially Philippine science. I won't say that we are lagging because we are already, I'm already allergic to the term we've been saying we're lagging. No, I won't say that. I think we are now developing our own, our scientific community. We're gearing towards developing our own science, local science, and increasing our capacity as well. So I think each one of us has to contribute in the developing and pushing the wheels of science, and not to stop learning. And I would encourage someone out there to explore bioinformatics, molecular biology, and contribute to genomics and transcriptomics, and even epigenomics. Thank you.
Thank you very much, Sir Nelson. Can't stop, won't stop, shouldn't stop. Okay, thank you. Ma'am Lillian, your message, ma'am.
Yeah, thank you. First, thank you for inviting me to be part of this event, and I learned a lot from other speakers as well. And for my message, let us continue to contribute worthy science that will solve solutions and improve the lives of the farmers in the Philippines, because they need to understand also the basic science and the basic science that we are doing that will help them to improve their livelihood, and also to address the food supply as well as other health concerns of our livestock animals. And also, I would like to encourage the young young researchers and also students to also involve in cryobiology cryopreservation, we need to basically we need more genetic resources to back up our important, especially the local genetics that we have in the Philippines. Thank you very much.
Thank you very much, Ma'am Lilian. And indeed, if I may parang borrow from one of your aspects, diba? Science should be done for the people. That's why we're doing it, in service of our countrymen and the people. And yeah, that's a noble thing to put as a foundation why we're doing science. Diba? Not just to sate our own curiosity, but also to help find solutions to pressing problems in the country and also in the world. Yeah. Okay. So with that, we're closing our Q&A forum. Thank you very much to our speakers. Don't turn off your videos yet. We're going to proceed with the awarding of certificates, as a symbol of our appreciation. Okay, to all. Okay, so let me first read the body of the common body of the virtual certificates that we'll be handing over, and then I'll be virtually giving it one by one to each of you. Okay, so let me read the body, the text. Okay, so the Philippine Genome Center awards the certificate of appreciation for being a speaker during the Genomics and Bioinformatics Conference, Overcoming Challenges, Building Opportunities, on December 8, 2021, hosted from A. Maria Regidor Street, PGC Building, University of the Philippines, Diliman, Quezon City, 1101. Signed by the Executive Director of the UP Philippine Genome Center, Dr. Cynthia P. Saloma, and yours truly, actually as Program Director of the Core Facility for Bioinformatics, as well as the head of the Program Committee. So this certificate is awarded to Dr. Heidi F. Galvez, okay, for giving us the keynote speech for today's this day. Okay. Okay. So Ma'am Heidi, this is your certificate. So next up.
Yeah. Thank you.
Yay. Okay. Next up, okay, is to this certificate of appreciation is awarded to Dr. Nelzo C. Areful for being a plenary speaker and a research speaker for this today's conference proceedings. So Dr. Areful, here's your certificate. Thank you very much. Thank you very much for your input and your expertise. Yay.
Thank you.
Right. Next up, okay, you have to Dr. George Hill Angeles. I hope I pronounced that right. For being a plenary speaker. Yes, I got it. Okay. For being a plenary speaker, resource speaker for today's conference. So, sir, thank you very much. Thank you very much for your time. Thank you for your input. Okay. And last but definitely not least, okay, to Ma'am Lilian T. Villamor, okay, for also being plenary/resource speaker for today's events, activities. Okay, so ma'am, here you go.
Thank you very much.
Thank you very much for your input. Okay, so of course, after the awarding of certificates come the photo opportunity. So that's why I asked you to keep your videos on. So guys, from the back, Ms. Rachel, are you there? Or
yes, sir. Please count down and we'll take the screenshots.
All right, so get ready. So here we go. In 3, 2, 1, cheese.
All right po, one more po. 3, 2, 1, smile. Yes, all right. Okay na po, salamat po.
Okay, thank you very much, Rachel, for taking our picture. And thank you very much to all our speakers for the time and for your inputs and your expertise. And for, of course, sharing your knowledge and your insights to our audience regarding opportunities and challenges in agriculture, livestock, and fisheries. Okay, so with that, okay, we end our activities. Thank you very much, everyone. If you have more questions about the conference, you could email us via capacitydevelopment@pgc.up.edu.ph, or you could visit our website. Okay, that's the URL is pgc.up.edu.ph. Okay, and also we would like to remind everyone to check the link to the feedback form in chat and also posted on the screen, right? That's the bit link, okay? This survey form will serve as the reference for the registrant's attendance to the conference, and it will be open up until 6 p.m. today, right? So please make sure to type your full name correctly, as certificates of attendance will be generated based on the form response. Okay. All right. So with that, thank you very much, thank you very much, everyone, on behalf of the program committee, and also with the co-host, actually the main host, Ms. Shalette Monzon. Okay, that is it for day 2 of the PGC Genomics and Bioinformatics Conference, Overcoming Challenges and Building Opportunities. This is not the last day. There's still day 3, which is set for tomorrow. Okay, same time, same channel, or same Zoom room, if you're, this age. Okay, with that, okay, we're closing today's activities. Thank you very much. See you tomorrow. Bye-bye.
Bye.
Bye-bye.
Bye.
Thank you. Bye-bye.
Bye-bye.
Thank you. Bye.